So our speaker today is Dr. Yevgeny Brudno. So by this time, I think many of our senior faculty members have met Yav, and some of our medical school faculty members and some of our students also met Yav. But for the rest of the audience, please give me a few minutes. I want to introduce Yav to all of you. Yevgeny Brudno, currently he is a Wyss Institute Fellow and at Harvard. And Yav did his bachelor degree and bachelor degrees in chemistry and physics from University of Pennsylvania 2004 and received his PhD in chemical biology from Harvard. And he is currently developing the first technology for non-invasive replacement of drug growth factors using a very fantastic platform. And in parallel, he's also considering commercialization of those technologies. And back in his PhD studies, he developed this unique method which can rapidly synthesize and screen sequence-defined synthetic materials. So what impressed me about Yav's work is he really come from materials, chemistry, chemical biology background, but have done a lot in translational research and moving forward this drug delivery area into really combine the platform with arbitrary combination of the drug screening in the real time. So in the recent years, Yav has been dedicating on developing the wide applicable method which allowing for the repeated and local and the very controlled release of therapeutical factors and those different chemical cues. Okay, so the rest of the 45 minutes we'll listen to Yav's story. Okay, let's welcome our speaker.
Hi, thanks so much for having me. I've had a really fantastic 2.5 days at Dartmouth, meeting with a lot of the faculty members here and the students, and so thanks particularly to John and Carl and everyone else who was involved in the process of setting this up, as well as Louise for really being fantastic, and I'm sure what was a very difficult schedule to create. So as Dr. Zhang mentioned, I'm Yevgeny Brudno, and I'll tell you about some of the work from my postdoc and my institute fellowship on sort of novel ways of delivering therapies to sites in the body. And really what motivates me when thinking about these things is thinking about a lot of the inefficiencies in current drug administration, or at least the traditional ways in which we have given drugs for centuries now, which generally target systemic, the entire body systemically. The problem with these methods, whether they are given orally as pills that you might take, or IV infusions for a lot of the biologics that are coming down the pipeline now, is that only a small fraction of the administered compounds actually end up at a site of disease where you might want them. This then requires very large doses to be given, and in the cases of chemotherapeutics, doses that are close to the maximally tolerated doses of drugs before you see significant toxicity. Many of these compounds seek rapid clearance or degradation from the circulation, and that requires repeated dosing. IV infusions are pills that are given daily or weekly. There is significant processing of these compounds that often leads to inactivation of drugs or the creation of toxic metabolite. And these three things combine to create, in many cases, off-target or systemically toxic side effects that make a lot of treatments difficult. And anyone who's had relatives who have gone through chemo or have been on immunosuppression knows that we can do a lot better in terms of sort of delivering these kinds of drugs. The lab that I joined after my graduate work was really focused on developing methods for local drug delivery. And these are implanted scaffolds, gels, stents, many beads, many other systems that slowly and locally elute drugs. So you get tunable release. the therapy is localized to one area. The release can be over a long period of time, and you don't have the spikes and troughs of systemically administered systems. And importantly, these releases are often tunable. So if you want very fast release, you can do that. And if you want considerably slower release, you can get those kinds of systems as well. And all of these combine to give you reduced systemic cyclicity. So I'll tell you two stories about how we've sort of taken advantage of this system and then actually combined local drug delivery with systemic administration to do some new and, I think, very cool things. So the first story really revolves around therapeutic angiogenesis. So therapeutic angiogenesis is the process of engaging different types of cells that are components of the blood vessels into growing into tissue that is currently lacking oxygenation. So if a patient has had a stroke or a heart attack, or perhaps simply patients with blocked vessels leading to lower limb ischemia or peripheral artery disease. All of these diseases are poor blood flow to a specific area of the tissue. And so what you really want to do in that specific tissue is sort of encourage the growth of new blood vessels, or angiogenesis. So really this work talks about engaging different types of cells, endothelial cells, as well as perivascular cells that sit on capillaries called pericytes, and delivering different growth factors to improve vascularization at these sites. So here is a very oversimplified kind of model of how these different cells interact with different growth factors. So whenever you have ischemic or low oxygenated tissues, these tissues start to express a lot of pro-angiogenic growth factors. Here I just outlined VEGF and angiopoietin-2 as very important ones. These factors create gradients that encourage two things to happen. They encourage endothelial cell sprouting, so the lining of the blood vessel to sort of sprout and move towards the ischemic tissue. And they also encourage perivascular cells that sit on top of these endothelial cells to detach and migrate away from the endothelium. These pericyte cells are actually blocking endothelial sprouting. So first you get the pericyte detachment and then the endothelial sprouting. As you're forming these long endothelial sprouts, they actually release various factors, including PDGF, that recruit perivascular cells back onto these sprouts. And through Ang1 related signaling, you have the maturation of immature vasculature into well-matured, luminized vessels. So as a postdoc in the Mooney lab, I started to develop some in vitro models for studying this process of pericyte and endothelial cell interactions. Now, there are many in vitro systems for studying endothelial sprouting, and this is just one example that I show here. As you can see, these endothelial cells are sitting on a bead, and then they start to form these nascent sprouts, and then they actually develop into multicellular structures. But there were many, many fewer in vitro models that actually incorporated the function of these perivascular cells, and that's sort of what I set out to do. And so what I show, for instance, in this movie is that you start out with a dextrin bead that's coated in both endothelial cells, which are red, and pericyte cells, which are green, inside of a fibrin matrix. And in response to various growth factors, what you can see is endothelial sprouting and pericyte migration. Here you can see just the pericytes migrating away into the fibrin gel in response to these factors, whereas these behaviors are not seen in the absence of these various growth factors. And we can start to quantify these behaviors. We can quantify sprouts, both in the presence, in red, and the absence of pericyte cells. And we can even start to get some interesting things, such as the fact that pericyte cells, as expected, decrease endothelial sprouting, which is what they do naturally on a capillary, but actually they decrease VEGF and Ang2 supported sprouting in very different ways, which was quite interesting to us. Most importantly, though, sort of looking at sprouting, is that we see that if you give two important factors for vascularization, angiopoietin-2 and angiopoietin-1 together, you essentially abrogate the entirety of the effect of angiopoietin-2 for these sprouting events. And I mean, this was previously known in the literature because they bind the same site and they actually compete for the same cell surface receptor. We can also look at pericyte migration away from the endothelium. And what was really interesting to me is that the combination of VEGF and PDGF actually inhibited pericyte migration compared to VEGF. And this has some parallels in the literature as well. It seems like VEGF and PDGF inhibit each other's activity when given simultaneously. So if they're incompatible, if we can't give them simultaneously, what can we do? Well, we have to give them in some sort of spatially, temporally regulated way. And so here is just a couple of different temporal regulations. This is, once again, endothelial sprouting, and we can sort of quantify and we can give different amounts of VEGF angiopoietin-2 or PDGF angiopoietin-1, either nothing the entire time in a sort of increasing way, in a decreasing way. These are days, and this is sort of concentrations, with a constant concentration, or one in which VEGF is high at the beginning, goes lower, and PDGF angiopoietin-1 is low at the beginning and goes higher. And in endothelial sprouting, that really gave us the best results in terms of seeing the most sprouting, which makes sense considering that a lot of these factors are incompatible. So now that we have some of these in vitro models and we've learned a few small things about how we might want to space these growth factors, another graduate student in the lab and I did some in vivo work where we looked at systems for incorporating growth factor into scaffolds that are implanted subcutaneously in such a way that either the growth factors are part of the polymerization process and end up in this macroporous network close to the pores of the scaffold, or if the growth factors are pre-encapsulated in these PLG spheres and PLG particles and then made into a scaffold, they end up being farther away from the pores. And what this does is you can have some of the growth factors close to the pores, and then they will release very quickly. And then some of the factors can be much farther from the pores, and then they will release much slower. And what Alessandra did was sort of characterize both the in vitro and the in vivo release of these systems, showing that you can differentiate getting rapid and intermediate release of our growth factors. So then you implant these scaffolds subcutaneously, and you start to look at the vascularization that takes place. And so here, for instance, we can look at the number of microvessels that grew into our scaffolds. And we can show that the combination of VEGF and angiopoietin-2 gave the best results in terms of the number of microvessels in our scaffolds. And if at the same time you try to give these PDGF and angiopoietin-1 molecules, you actually significantly reduce the number of microvessels that grew into our scaffolds. However, if you had the rapid release of VEGF and angiopoietin-2, these pro-angiogenic molecules, and the delayed release of PDGF and angiopoietin-1, you could abrogate some of this inhibition to the point that these two were not statistically significant in comparison to each other. On the other side, instead of looking at the vascular system, you can look at the number of pericytes and smooth muscle cells that are now lining our vessels. And you can characterize the number of alpha SMA positive vessels in these scaffolds. And once again, this delayed release of PDGF and angiopoietin-1, which had the better endothelialization, the better vascularization, sort of also created improved numbers of what we think of as more mature blood vessels, the alpha SMA positive blood vessels in these scaffolds, suggesting that delayed release of these factors recruits the kind of perivascular cells that we want sitting on our endothelium, but sort of doesn't inhibit the initial formation of those vessels. We can also look at vascular remodeling over time. So we can look at 2 weeks or 4 weeks after the implantation of our gels. And sort of here is where ENG1 really shines as a factor. Because early on in the 2-week period, what you really see is a lot of very small capillary-sized vessels and very few arterial-venule-sized vessels. And especially in the VEGF angiopotent 2 case, that really doesn't change very much from week 2 to week 4. However, once you start to add ENG1, PDGF, and most spectacularly, when you add the combination of PDGF and ENG1, you really see a vascular remodeling event, the maturation of these vessels to more arterial and venule type systems, which is something that we had hoped to see. So the conclusions from this work really is that in some cases, and specifically in the case of therapeutic angiogenesis, you really want to separate in time the presentation of the pro-angiogenic and the pro-maturation factors. And that this really does provide better conditions for neovascularization and angiogenesis, at least in our models. And these pro-maturation factors are absolutely critical, but they do inhibit the function of the pro-angiogenic factors that if they're given simultaneously. However, when you give them separately, you improve the recruitment of these perivascular cells, you improve the vascular remodeling when it's given in a delayed way. And so, I think the big takeaway is, some of these in vitro models can be very powerful in understanding and recapitulating some of the in vivo cellular behaviors that we're seeing, and so can really be used for a lot of different systems. Now, something that I'm really excited about taking this further is the possibility of testing additional factors and peptides and small molecules, to really look at their impacts on pericyte function, especially in our in vitro work. And, really studying how growth factors modulate pericyte recruitment and other interesting systems in vascularization. Finally, sort of some unpublished work that I have actually focuses on this pericyte recruitment. Now, in the past, all the data I've shown you focuses on pericyte detachment. This is actually the recruitment of these green pericytes in this gel towards this endothelial ball, which is endothelial cells that are on the surface of a dextrin bead. And what you can see in full media is that these pericyte cells really in a directional way migrate towards the endothelium and really aggregate here. Whereas if you block PDGFR, the receptor that's commonly thought to mediate this recruitment, then you still see the migration of the pericytes, but you don't see the directional migration towards the endothelium. And so I think there's a lot more to be done with these kinds of models in understanding pericyte recruitment. So let me take a small step back and sort of come back to this first slide that I had, when I talk about systemic drug administration, and just think a little bit more about this local drug delivery and whether it has any fundamental limitations. And one limitation that I sort of felt constrained by when I was doing this pericyte work is really that there was no opportunity to refill our gels once we had released all of our growth factor. And, if I go back just a few slides, I can show you, when we were looking at these release factors, in the in vitro release case looked very good. But when we took these systems and took them in vivo, we saw that even our intermediate release was relatively rapid. And so the question became, well, how do we get a system that, say, truly releases something and then, say, 24 hours later releases it again, and 24 hours later releases it again? Or potentially, what if you wanted to release a different drug at a different time based on how the disease was progressing? So if that drug was killing your cancer, you would keep giving it. However, if the cancer was not responding, you could switch out the drug and now locally deliver a completely different drug. So how do you do true temporal separation, change the drug? And all of these systems, in my mind, really necessitated taking these things and being able to refill them with new components. And this is what we sort of came up with So here is let's say a patient that has an intraperitoneal tumor. You would take a gel or some sort of injectable material and implant it close to or even inside of the tumor. This gel could be, to start, filled with a drug that slowly releases and to some extent decreases this tumor. But then we have this gel be modified in some way so that when we gave a systemic administration of drug, IV infusion, oral administration, and this drug circulated throughout the system, it specifically homed to or targeted or bound or was captured by this gel and thereby got refilled with more of this drug. Once the drug was cleared from the systemic circulation, this gel was refilled and ready to go for another round of slow elution. And so the question became, how do we get minimally invasive refilling of drug depots and allow repeated, controlled, and local presentation of drugs or clinical compounds, at specific disease sites? And I have two different systems, what we call Generation 1 and Generation 2. Generation 1 really takes advantage of nucleic acid recognition. So you could conceive of this gel that's sitting here releasing a drug, but also modified with single-stranded nucleic acids that have been chemically modified to be stable inside of the body. After this depot is exhausted and there is no more drug, you can think about giving something like a nanoparticle that's carrying complementary nucleic acids. These nanoparticles are going to circulate through the blood. They're going to extravasate at sites of penetrance, such as at a tumor. And they're going to get bound up in a sequence-specific way just through the recognition of these nucleic acids. And now these nanoparticles are going to be held in position as they release their chemotherapeutic cargo or their drug cargo of some form. And so that's the first thing we went into after. So I made up some of these alginate gels that we really like. These are calcium cross-linked alginate gels that carry these nucleic acid strands. And then I can take some fluorescently modified nanoparticles, modify them with complementary nucleic acids or non-complementary nucleic acids as controls, and sort of just in vitro incubate them together. And you can see if the gels and the nanoparticles are complementary, the nanoparticles bind, and the nanoparticles are fluorescent, so you can see that. If you have non-complementary controls, you really don't see any binding, and we can quantify these kinds of systems. So this works well in vitro, and now we can go in vivo. So we had B16F10 melanoma-carrying mice, which were injected intratumorally with a gel that was carrying this nucleic acids. As control, we had gels that carried a different sequence of nucleic acids, or mice that were never injected with gel in the first place. And then we gave all the mice retroorbitally IV injection of these nanoparticles that carried the complementary nucleic acids. And so what you can see, or what we saw, or what I saw, looking at these fluorescently labeled nanoparticles over time, is that on day one, there's actually equal amounts of these nanoparticles in the tumor, no matter whether the tumor had the complementary nucleic acids or the non-complementary or even didn't have the gel. And that's because these nanoparticles are getting in through the enhanced permeability of the tumor. But as you look over time, you see that the nanoparticles are resident at the tumor site for extended periods of time. They are being bound up in the gel. There's maybe even a small increase in the amount of them in the gel. And then they are residing at the gel site or in the tumor for significantly longer than nanoparticles that can't bind to the gel because it carries non-complementary nucleic acids or because the gel isn't even there. So we're extending the amount of time that the nanoparticles are retained at the tumor, and therefore giving them a longer opportunity to release their cargo. So we can then take this into a therapeutic model in a MDA MB231 breast cancer xenograft model, which are intratumorally injected with gel on day 0 after growing up for a couple of months. And then we can give IV administrations of doxorubicin-carrying alginate nanoparticles at 2 weeks, 3 weeks, 4 weeks, and 5 weeks post-gel injection. And, with the same amount of drug in control versus the targeted therapy, we really see the inhibition of growth for our targeted therapy and the continued growth of the tumor in the control cases. And we've done a number of different controls where the only difference was whether the nanoparticles could home to the tumor, but also some bolus doxorubicin controls where doxorubicin was not given in a nanoparticle formulation. And in all cases, our targeted system outperformed the non-targeted or the bolus doxorubicin systems. So this was really exciting to see, especially when you start to compare sort of what happens right after the administration, and our targeted therapy reduced the tumor size, whereas the controls still continued to grow. This DNA-based system is actually also a great candidate because you keep using up the DNA sites on the gel. There is a theoretical limit to how many times you could potentially refill. But through some clever DNA nanotechnology work that was done, that was originally published elsewhere at the Wyss, and really got me excited, we started thinking, well, this actually doesn't have to be a limitation. And together with Elliot Chaikov at Beth Israel, we started thinking about refilling the surfaces of vascular grafts, and specifically small vessel grafts that really suffer from thrombosis. So the Chaikov lab has worked for many years on bypass grafts that are bypassing some sort of plaque or a clot. And they coat these grafts with any number of different anti-thrombotic agents, but in all cases, their anti-thrombotic surfaces wear down over time through the action of proteases and nucleases that are present. And so we thought about, well, if you had a surface that's modified with these DNAs, you can take advantage of something called toehold exchange to replenish these surfaces. And so how that works is, well, for starters, you can just load them up. So here is our anti-thrombotic agent, and now it's loaded onto the surface. Once again, we're using chemically modified nucleic acids that are not degraded by serum. And then over time, these, say, the Chaikov lab specifically likes a protein thrombomodulin, will start to get degraded. So how can we pull these off and put in new ones? Well, we use toehold exchange. So we take another strand that's complementary to a different part of the nucleic acid. Up. So they come down, they sit down in the part that's open, they replace the previous nucleic acid, and those nucleic acids falls apart. And this is an equilibrium system, so you flood a vessel with one type and it will completely displace the other, and then you can actually do the reverse of this. You can flood the system with the top ones and they will remove the others. And so that's sort of what we've done in serum. This is still a work in progress, but we can sort of take, this is fluorescent streptavidin proteins that are on these nucleic acids, and we can sort of replace them back and forth over time. So we can keep replacing these systems, and this is actually in serum that we're seeing these effects. Moreover, we can do some kind of things. TBA is an antithrombin aptamer that we use, and so this is sort of the inhibition of fibrinogenation that we see. If we just have nothing, there's no inhibition. If you just have this antithrombin system, but it's not linked to the surface, you don't really see any inhibition. If you just have the surface nucleic acid, that's not really doesn't do anything. This combination of the antithrombotic strand and this nucleic acid, really gives you inhibition of fibrinogenation. Then you can take an enzyme, you can chop this up, you can eat this up, and once again you lose, if you treat it with T5, you completely lose the activity of the antithrombin. And then you can take it, you can replace the strand with the new antithrombin, and once again you have inhibition of fibrinogenation. And now we've done this through a few cycles, and we're preparing this work for publication. So let me now tell you about the other system that we have. And this one really came about because we felt that these nanoparticle systems could be very useful in cancer, but really we were excited about a lot of other diseases. And really what that necessitated is going into small molecules. We got excited about antimicrobials or immunosuppression and started thinking about what would it take to really make this a translatable technology. And one of the major pieces of feedback we've gotten is that nucleic acids are too large and that nanoparticles oftentimes don't extravasate into the tissues where you might want them to use. So we really wanted to come up with a small molecule-based system for doing the same thing that we had done with DNA. And we really focused on bioorthogonal click chemistry as one possibility to have a gel with binding sites, bringing in compounds that sort of refill these gels and then release drugs. So here is just one sort of example of this. So these are mice that have received ischemic leg injury. It's a ligation of the femoral artery. And so at the time of ligation, we also inject a gel into the muscle intramuscularly. The gel can be either a control gel or carrying this chemical motif, the azide, which specifically reacts with cyclooctines. Now then, after that, we give IV injections of a fluorescent probe that is carrying the cyclooctine and is expected to specifically bind with azide. And 24 hours after that, what we can see is that it has specifically targeted this muscle, whereas a control gel that didn't have this chemical motif does not have this kind of concentrator effect, and we can sort of quantify that. We can look over time and see, so for instance, we can keep adding this fluorophore and keep getting increases in the limb targeting effects that we have. This is sort of an artifact of the imaging that it sort of spreads out this way. The true sort of location of the fluorescence is actually centered on this. This is kind of an artifact that you see with IVIS imaging oftentimes. We can really quantify how much we're getting. So the gel is only 0.5% of the total tissue, total body of the mouse, but we're seeing about 6.5% of the injected dose captured at the gel site. So we're really concentrating this systemically administered drug at this very small gel site. And this 6.5% of the administered dose is actually only about 0.05% of the available sites on the gel. So suggesting with this administered dose, we can refill as many as 2,000 times. Although for some of our chemotherapeutic work, we have increased the dose, thereby decreasing the number of theoretical refills that we can do. But there's multiple chemistries that we can think about using, multiple chemistries that have been published in this general scope of bioorthogonal chemistry. So the one I've been talking about is the reaction of azide with this cyclooctine motif. And that has worked well, but we can also take tetrazine motifs that are known to specifically react with transcyclooctenes. And if you have a gel that's carrying an azide and a gel that's carrying a tetrazine, and they're injected in two parts of the mouse, one intramuscularly and one intramemory, we can then give a mix of two compounds, mix them 1-to-1, and give them IV, and see one compound localized to this region and a second compound localized to this region, and we can quantify that. And the silly example that I always give to this is, let's say you have an elderly patient who suffers both from cancer and from ischemic disease or peripheral artery disease. It is entirely possible that you want to promote angiogenesis at one disease site, but you want to inhibit angiogenesis at the cancer site. And currently, there's really no drugs that would let you do both things at the same time. How do you promote something in the leg and inhibit it at a tumor site? But potentially something like this would really let you do incompatible systems at the same time to different locations. I've gone on and shown that you can orally refill. So this is once again a mouse that has an intramuscular gel, but the fluorescent compound is now given orally. Through oral gavage, the mice internalize it, it circulates in their blood, and concentrates specifically at the gel site. And we can sort of quantify that. Finally, now that we're thinking about chemically modifying a drug such that it is carrying a targeting motif, the specific click chemistry targeting motif that lets it bind to the gel, why not use that opportunity to create a prodrug? So the combination of drug, the cleavable linker that releases the drug, and the targeting motif would be something that would be non-toxic, that was incapable of crossing the cell membrane, and therefore wouldn't have any effects on its own. So what's circulating systemically is this non-toxic prodrug that has no effect, except that it is able to be captured by the gel. And so at the gel, it's accumulating. The rest of it is excreted from the system, excreted from the circulation through kidney filtration, and you pee it out. But over time, this cleavable linker releases the drug, and it releases it far slower than the time it takes for the drug to be cleared from the body. So the only place where this prodrug remains is at the gel site, and that's where it's slowly, the linker is cleaved and the drug is released. So you only really see the drug locally near the gel site. And so that's where we're going now. I have considerable preliminary data creating some of these doxorubicin prodrugs where we take doxorubicin, we make cleavable linkers. In this case, this is a hydrazone that will hydrolyze with water into the ketone of doxorubicin, and we'll link it to these cyclooctane motifs necessary for click chemistry. These kinds of materials are significantly less toxic. So if this is concentration of our doxorubicin, and this is the viability of our cells as measured by Alamar Blue assays, so really the metabolic activity of the cell, what we see is with doxorubicin, we see the reduction in cell metabolic activities at concentrations that are orders of magnitude lower than for some of my best prodrugs. So this prodrug 1 is this compound right here. So now, we're making things that are considerably less toxic, but can still release active, naked doxorubicin with different kinetics depending on the pH of the tumor. So at the pH 7.5 that you would have in the blood, really the half release is at over 150 hours, and most of this drug is gone from the circulation within about 12 hours. But if it's there for a long time, and especially in the acidic environment of the tumor, this drug will be released locally at the tumor site. So we're really excited about this work and about some other potential work. We have a bunch of collaborators. We're sort of thinking about this for targeting resection margins in cancer therapy. So the idea there would be you remove a tumor, you do a tumor resection, you leave a gel behind, and this gel is repeatedly refilled and releases chemotherapeutic in order to get at the margin cells that you might have missed. We're thinking about this as refillable depots for some of these ischemia issues like myocardial infarct that we're sort of excited about. As I mentioned, we're working on refillable drug-eluting vascular grafts and vascular stents, as well as anti-thrombotic surfaces for vascular catheters. And with Jim Collins at MIT, in work that I haven't had a chance to tell you about, we're thinking about targeting implant-associated infections. So modifying the surfaces of implants in such a way as to be refillable with antimicrobials for these really devastating infections that people have post-implantation. So, I think in terms of where we're going with this, I think, we've been commercializing this technology for about a year now, and we've really identified a couple of key things that, in terms of what diseases would really benefit from this. I think you really want localized disease. You want disease that would benefit from repeat treatments, and diseases that are currently managed with dangerous or invasive procedures. And that's where we identified liposarcoma, some of the oncology applications. I think this does have wide application in many diseases. And, the future work really is in prodrug therapeutics, the potential of prodrugging immunostimulants for modifying the tumor and microenvironment, and prodrugging nucleic acids, for instance, for siRNA delivery are some of the things that I personally am very interested in. So I have a lot of people to thank, but I hopefully will go very fast. So, I've been just incredibly lucky with the people that I've gotten to work with and for. So did my postdoc with Dave Mooney, and he's continued as my academic mentor for my Wyss Institute fellowship. And there's been a huge number of people, Professor Neil Joshi and Rajiv, who's a joint student between Neil and David, really, have a lot of experience with the click chemistry work that I've really benefited from. Brian has done a lot of the ischemia work. Alex and Stephanie and Matt are people that have worked with me. Alex was an undergraduate, Stephanie a rotation student, and Matt is an animal tech that have been just utterly phenomenal. And Eduardo Silva is a graduate student who got me started in the lab when I first got there. The Wyss has been fantastic. So these are people that are part of the Wyss Institute. Michael Eisenberg is my industrial mentor as part of my as part of my fellowship. Ed, my sort of the platform leader, and a lot of these people, Chris, Mike, Sarah, Katie, have helped with animal work, materials characterization, materials development, and have really been fantastic. Finally, we have a really amazing BDIP team for commercializing this technology that have really just been incredible. So, and thank you to everyone here for coming to the talk and for your attention. Sure. So the sort of the question being, do these gels degrade quickly? How long do we think they degrade for? And so these are calcium cross-linked alginate gels that the Mooney Lab has used for at least a decade now, and we actually have a pretty good sense of their degradation and the kind of chemical modifications we can make to them to either stiffen them up or significantly increase their degradation. So the current system that we uses unoxidized alginate gels, high molecular weight. So they are designed not to degrade. And I think one of the slides that I showed you had gels in tumors out to 140 days. And still we can do click-mediated refilling after 140 days of the gel inside of the tumor. So these gels are really made not to degrade. However, if we ever wanted to control their degradation, we can start either lowering their molecular weights or oxidizing them. And we've shown that this really creates a biodegradable system that will over time degrade. So really, it's a tunable system. We've looked out 140 days. Unfortunately, after that, the tumors got too big, and so the mice had to be euthanized. So I don't have any data beyond 140 days, but we feel comfortable that if they're good after 140 days, they'll likely be good 360 days, but I don't know beyond that. Thanks.
So I'm wondering about size and shape and diffusion distances. So you mentioned you could attach it to stents, maybe that could be thin sort of film. You can inject these gels. What sort of size and shape can you inject, and what sort of control do you have over that and how it relates to your localized source?
Right, so you're sort of asking about when we do this gel injection into the tumor, what does the gel look like inside of the tumor, right? Is it a ball? Is it permeate throughout the tumor? We've tried a couple of things to get at that question, right? So we've sort of tried micro-CT. We were hoping that the high calcium content of the alginate would do it. We've sort of even thought about barium cross-linked alginate gels for micro-CT. We haven't pursued it too much. It's a very important question, especially, as you alluded, the diffusion of the drug is going to be a certain distance away from the gel, right? So I think I haven't explored it very much. These are also animal models, that aren't necessarily predictive of human tumors, right? Which I think many of which, different tumors are also very different in terms of the diffusion through them, especially working with some of these melanoma, which are very liquidy. as tumors go. My suspicion is that they are not pure spheres, you know. The gels are injectable, they are shear thinning, and the tissue we're injecting into, especially with these MDA and B231 tumors, is actually fairly stiff. So my strong suspicion is that when we inject, it does permeate the tumor to some extent, but I've never gotten a good sort of understanding of where do they go and what do they look like. And I mean, you know, I'm sure there are imaging modalities that perhaps, you know, we've thought about that they're too big for 2-photon and, you know, too deep for 2-photon, and the micro-CT didn't work out as well as we would have liked. But I'm sure there's something out there that we could use, maybe barium cross-linked allogeneic and micro-CT. I'd love to explore that space more. Yeah, so I think what you're referring to is sort of the foreign body response that often accompanies implants or injected depots at various parts of the body. You know, we have done histology on these gels inside of the tumor after, you know, hundreds of days, and we don't see much of a fibrous cap around them. We see a fibrous cap around the whole tumor. And that's just, I think, part of the animal model. But we haven't really seen as much, you know, in that respect. Certainly, if there is a foreign body response, it will significantly reduce the amount of drug that is capable of getting to the gel, right? I mean, that is related to the vascularization of the tissue around the gel. So it's an excellent, excellent point. I think we need to explore it further, but we haven't seen a lot of foreign body responses. And I mean, we found that to be quite comforting. And it fits with the general literature on ionically cross-linked allogeneic, that they really don't elicit, especially if you use like really clean allogeneic. The ultra-pure material doesn't really elicit a lot of foreign body response.
Yeah, I want to follow up with
Yeah.
So the refillable allogeneic gels, I'm understanding you still require systemic dosing to reload and refill the gel, correct?
Yes.
So what can you tell us about, I mean, that seems to me that gives you the opportunity to control side effects by reducing the dosage relative to a strict therapeutic dose. And so can you tell us anything about relative dosing? And do you have the opportunity to play with the relative rates of absorption versus clearance? And so you can systemically dose at a much lower level, refill essentially much more slowly, and stay below any toxic threshold of the body.
Yeah, so let me separate your question into two parts, if I may. So the sort of this question of could you lower the dose, right? Let's ignore the prodrugging for the second, right? Let's just talk about sort of native toxicities, especially in the nanoparticle field, right? I think we have one of two modalities. What it looks like is at the same dose, our material is more efficacious, right? You could turn that on its head and say, well, at a low, we can have the same efficacy and have a lower dose. I don't know which one of those two I would prefer. Do you want to have the same side effects as you're currently having and just have it be more efficacious? Or do you want the same efficacy? I think given current cancer outcomes, which in most cases is a couple of months of extended life, I would prefer equal side effects and improved efficacy. Now, perhaps in certain cancers that are curable, so Hodgkin's lymphoma, right, or childhood leukemias. Now, those are systemic, so that's not a localized disease. But for some of the localized curable cancers, like certain HER2-positive breast cancer, there I would say they're curable. So the efficacy is good enough. So let's reduce the side effects. In cancers that are giving you 2-3 months of extended life, I would say screw the side effects, improve the efficacy. Now you bring in the prodrugging aspect of it. So now I'm saying you can significantly reduce side effects and increase dose, right? So then I say increase the dose because you're reducing the side effects. Not, don't even keep the dose the same or decrease it and have the better efficacy.
I kind of follow up on the release part of this research. Is this refillable debody very strong idea? Once you refill the debody, and many of the gels, as you know, are responsible for many chemical cues and even for light. So in terms of releasing the different pattern, have you thought about different mechanisms you can further develop the gel into the way of not only deliverable, but releasing the way of programmable?
Yes.
Temporary spectrum.
Yeah, so what you're getting at is sort of combining the refillability of depots with a lot of the work that has been done for on-demand drug release, sort of by physical cues, ultrasound, light, or even local magnet, or prostate surface antigen, right? Like prostate specific PSA, or like local enzyme therapy. So we've thought about it. I've thought about it. I think there's something there. Each mechanism is going to be different, So especially in the click cases, the covalent bond, right, is something you really have to take into account between the click motif and the drug. And so if it's not a hydrolysis, you have to cleave that covalent bond somehow. So, where we thought about the most exciting thing would be something like focused ultrasound, right? Because that goes deep in the body. But I don't know how to break a covalent bond with focused ultrasound, right? Something like light, I would worry about because it doesn't really penetrate far enough, right? I think that would be an excellent proof of concept, but I don't see that as necessarily translating into a therapy, simply because a lot of the light sources wouldn't be able to penetrate where you would want them. Now, PSA cleaved systems, MMP cleaved systems, those would be really interesting. And those would be really interesting for a second reason, that then that material could hang out for a long time, I don't know how long, but for a long time, until, say, tumor recurrence, right? So if you removed the tumor, you left the gel there, and you filled it or you refilled it, right? Then that stuff could stay for a long time, and as the tumor recurred, it would start expressing PSA or MMPs, digest these materials and release the chemotherapeutic right as when the tumor is most sensitive, right as it's only just getting started, right? And you could refill those. So something like that would be really interesting, both from a diagnostics and from a therapeutic area. Yeah, so I think that would be really cool. I haven't explored it.
So it looks like, I guess I may have when it comes to your renewable capture sites, your total competitive DNA technology, it looked like that there was kind of a progressive loss of efficiency by which that happened. First question, what's going on there? And then maybe a related question is there opportunity to leverage affinity capture and maybe tuning on rates and off rates of affinity capture of some handle in order to load gel and then have it dissociate lead what is kind of a truly renewable capture.
So the first part of your question was, why don't I jump to that slide? I know exactly why it's happening, so I can. So sort of what Dr. Grivel, you're sort of saying, well, it looks good at the beginning, right? But then it's sort of, why is it like that? And so this is done in serum, and the nucleic acids that Alex used, so this is together with Alex Miller, my undergraduate, used were actually native DNA. These were not chemically modified. These were when we were still modified. So the trick with these kinds of DNA nanotechnology systems is once you modify the nucleic acids, the rules for the kinetics change. So we were using naked, native DNA for these systems. What we are now doing is moving to mirror image DNA. And so what I'm hoping is we won't see this. But in serum, there are nucleases. And this DNA is just getting degraded at its edge with exonucleases, and it's really preventing these kinds of dynamics from happening. I think when we go to mirror image DNA, which unlike, they say, the 2'-methyls that we used in the in vivo case, don't change the DNA nanotech kinetics that we really require here, I'm hoping we won't see that. So your second question was?
Just maybe leverage or tuning affinity.
Right. So affinity, so like let's say you had an aptamer on a gel, and it sort of was able to capture a drug and then slowly release it. So there's actually a couple of labs that have done this. There's a lab at NC State, actually, that sort of uses VEGF and PDGF aptamers to capture them out of the circulation and then release them. There's a couple of challenges to that system. First of all is simply the number of binding sites you can put on a gel. These are fairly large molecules, and you know, the nice thing about the clicks is you can put tons and tons and tons of them all over the gel. The aptamers are much harder. Most aptamers, in the way they have been evolved, are native nucleic acids, RNA and DNA, and as I alluded to here, they would fairly quickly be degraded by biology, and there are very few aptamers that are chemically modified. So there is a VEGF aptamer that is, extensively modified in order to be serum stable. So you could use something like that. But for doxorubicin, one doesn't exist. There are these things called mirror image aptamers that people select for, which you could certainly use, but there's very few. They're called spiegelmers. And so the way that they do that is they select for the mirror image of the molecule, right? And so then the mirror image of the aptamer will bind your molecule. And so those things are very stable, but there's only 2 or 3 that have been reported so far, although that would be an interesting area. So I worry about the stability, I guess, of those kinds of systems.
So we have students, many students in the audience. So, if the students want to join your group, what type of students?
Excited about science? I think there's a lot of really productive and exciting stuff that can be done at the interface of, some of these drug delivery, biomaterial stuff, and the chemistry. Now, I don't expect any one student to, be able to cover that span. It took me 11 years to gain the training to at least even pretend to know what I'm talking about, right? But sort of, I think a lot of these things will have to be done in sort of groups or teams where, one person will be leading the effort, but we're going to have chemists and biomaterials people that are sort of, really have to rely on each other to make this work.
So both science background and engineering background.
Yeah, I think, I don't, quite know. I mean, everything will depend on but, so I think sort of kind of an interest in an interdisciplinary and kind of a focus on many different things. I mean, Carl understands this, right? Like, you can sort of have that kind of feel.